Hi there, my name is Tanner Toussaint, and I'm a junior in chemical and biochemical engineering with a minor in biomedical engineering. And the project that I was assigned to was the construction of a cell perfusion apparatus to model the dynamic 3D culture of the human trabecular meshwork. And just a little bit of background on glaucoma and the trabecular meshwork. Glaucoma is the leading cause of irreversible blindness in the United States, and the amount of affected people is expected to double by 2050. And IOP, intraocular pressure, is the only modifiable risk factor. And since it's controlled by the trabecular meshwork, this is what we focus all of our research on. So when fabricating the scaffolds to mimic the eye tissue when doing tests, there are four considerations. We have two different freezing types, -80 and then flash frozen using liquid nitrogen, and non-aligned and aligned scaffolds. And what alignment refers to is the alignment of the collagen fibers. And if you look at the SEM pictures here, you can see that the fibrils are aligned together in one direction for the aligned and not the non-aligned. And then this is a structure uh, that would essentially affect the proliferation of the cells. And also in my case, in my research, it would affect the pressure drop across the scaffold. So as far as methodology, over the first semester, our main goal was to achieve a steady state pressure drop throughout this system here. And as you can see in the apparatus, this allows us to have backflow throughout the system so that there's no air when running the transducer. And then over the second semester, once we achieved a steady state pressure drop using only water, scaffolds, and no cells present, over the second semester, we essentially switched the water out for low glucose DMEM media, and then we cultured cells and then seeded them onto the collagen scaffolds and put them in the bioreactor to see if there was a pressure increase throughout this apparatus. Some results from the first semester, when testing all of the freezing types and pore sizes, we found out that all of them had relatively the same pressure drop throughout the system. So this allowed us to use essentially whatever type of scaffold we wanted to use when measuring pressure increase throughout the system over the second semester. And since the non-aligned 80 scaffolds were working the best at other TM projects, we decided to start out with those. So some results when running the perfusion using a peristaltic pump for 24 hours shows that it was not enough time to get a steady state or steady pressure increase throughout the system using about 100,000 seeded HTM cells. And when looking at the 48-hour data, this is our best data that showed a relatively steady pressure increase throughout the system due to the presence of the human trabecular meshwork cells and the media throughout the system. And this was also done using a media recycle system. So we were able to do this for 48 hours at least. In conclusion, over the first semester, we were able to get repeatable steady state pressure data with a low standard deviation. And then over the second semester, we were starting to get data to show that HTM cells had a pressure increase throughout the system that would indicate it was a good model for glaucoma. And then just for reference, this is the setup of what the perfusion would look like with 12 reactors using a peristaltic pump. So if you guys have any questions, I would love to see them in the comments. And thank you very much for tuning in.